oxygen species. So, Patricia, the floor is yours.
Okay, can you hear me?
Yes, I can.
Perfect. So, can you see my screen?
No. Your screen is not visible yet.
Okay, got it. Now?
Yes.
Great. Well, first of all, thanks for the invitation. I was really glad and really thrilled when you invite me to all people that is organizing VAS. And also was very, very happy to hear the students organizations that is being actively participating. When I was a PhD student, I had the opportunity to attend to several GLEON meetings. It was very good for me. I met a lot of people, wonderful people that helped me through my career. Now I'm an early career researcher in Argentina, but in my early years, GLEON was a very wonderful experience. So, well, directly to my talk, I wanted to talk to you about natural levels and the photoproduction rate of reactive oxygen species, ROS, in different sub-temperate environments. My institute is in Idioma. It's an institute that research from ecological to plant to, it's a very multitudinal institute. I have my own personal webpage that you can visit, that is Patagonian Limnology, that you can find all of my updates and new things, and you have my access to my papers. Okay. As an introduction, I can tell you that in aquatic environment, the reactive oxygen species, or ROS, are important oxidative agents. The ROS can be found in the surface water of rivers, lakes, also are in seawater. They also, you can find concentration of ROS in the rain, in the dew, in clouds, in aerosol, and also in living organisms, because can be produced in the cells, inside a cell. The hydrogen peroxide, it's a type of ROS that is quite stable and it's long-lived, with a half-life that ranges from several to 100 hours. We make it a wonderful or useful indicator of oxidative stress because we can measure it. Other ROS, such as free radicals, they are very quick and they don't have a very long life, so we cannot measure it. So, in aquatic ecosystems, there are two sources of hydrogen peroxide. One is the abiotic source, and the other one is the biotic source. The abiotic hydrogen peroxide production can be the result of the photolysis of the breakdown of the dissolved organic matter, DOM. And the humid fraction of the chromophoric dissolved organic matter, which is the color dissolved organic matter that also has the potential to produce hydrogen peroxide, is the one that also prevents the penetration of light. So the abiotic production of the hydrogen peroxide is that we have the sun, we have the radiation, particularly the UV radiations break down the dissolved organic matter, and we have radicals, we have water, we have oxygen dissolved, and this is produce a very reactive dissolved organic matter, and also can produce, in some environments, can produce hydrogen peroxide. This is the abiotic production. This process is very light dependent, therefore it only occurs in the illuminated part, in the surface water. But there's also been known that in dark, there's also can be produced hydrogen peroxide. And this production is not dependent on the light, this is dependent on the organisms. So some bacteria, some algae, and some fungi can also produce some extracellular ROS in darkness. This is a very interesting topic because not all of the species can produce any ROS. There's very species-specific production. I don't think you can see my next slide. Can you hear me?
Yes, we can hear you.
Okay, you can hear me. Okay, just I think the
We can
internet is
also
not
see
so good.
live. Yeah.
Maybe it's just a problem of connection. Okay. In the southern part of Argentina, it is also known as Patagonia. It's a region that is shared with two countries, Argentina and Chile, and it's characterized by a different type of aquatic environment. That most of them are glacial origin. This is a map when we have South America. This is the southern part of Argentina, which is Patagonia, but Patagonia is also named in Chile. So we have different type of environments that include shallow lakes, deep lakes, streams, big rivers, and very big deep lakes. Okay, so as I said before, one of the substrates for the hydrogen peroxide production is the dissolved organic matter. So the first thing that we need to do is to characterize this dissolved organic matter in order to see if it can produce this hydrogen peroxide. Okay, so one of the goals of one of my last work was about do a field survey about the natural concentration of hydrogen peroxide in different aquatic ecosystems, different freshwater ecosystems. So for this, I also wanted to do some experimental incubation in order to see if this water can produce hydrogen peroxide. And one of my hypotheses was that there was a photochemical production and it was related with DOM characteristics and concentrations. So in order to assess these objectives, I performed a field survey in 12 different type of systems. The study area was the water samples were collected inside the Nahuel National Park. This is one of the first national parks that was created in Argentina. It's a very extensive area, but also has some cities and some settlements inside, because the national park was created after this creation of a city. So in this national park, we selected 4 streams, 4 shallow lakes that were less than 15-meter depth, and we also sampled 4 deep lakes that in average were 100 meters depth. Some of them, Nahuel Lake is one of the largest lakes in the area, and at the average it's around 200 meters, but it has been known that in some part of the lake there is 450 meters, so it's a very, very deep lake. So I'm just going to resume some of the materials and methods that we perform. We do some surface samplings from January to February 2018 using a camera bottle. Once in the laboratory, we sterilize the samples through filtrations. And we left an aliquot of 200 milliliters and for each sample site, and to perform very accurate characterization of the chromophoric dissolved organic matter, the fluorescence dissolved organic matter, and we also measure the dissolved organic carbon, the DOC. This is some of the equipment that we use that is available in our laboratory. And in order to measure the hydrogen peroxide concentration, we use a method that is scopolamine and horseradish peroxidase. It's a fluorometric method, and it was performed between 2 and 3 hours after the collection of the sample. It's a very fast method. And this method, it's chosen because it's very simple, it's very high sensitivity, it can detect very low concentration of hydrogen peroxide. We are talking about nanomolar concentrations, so we need a very sensitive methodology. Okay, briefly, the hydrogen peroxide did not fluoresce, so we needed to sort of stick it to the scopolamine, which is a very fluorescent component, and we're using an enzyme, the horseradish peroxidase, that oxidizes the scopolamine, and it is directly correlated to the concentration of hydrogen peroxide. So the decrease in the sample is proportional to the amount of the hydro, to the ROS. So after a sequential spikes of a standard, we can determine the actual concentration of the hydrogen peroxide. So we also perform some experiment, and in this experiment we collect the water from the field, and we assess also in these 12 study sites, and we expose the water samples to light, to radiation, to visible radiation, and also expose the samples to ultraviolet radiation. This was during 4 hours. We just put the samples to irradiate. We also have a control in darkness that was wrapped with aluminum foil, and with this we sort of perform a hydrogen peroxide formation rate. We just took the initial concentration, the final concentration, the initial concentration, and the time that was 4 hours, and so we have a sort of rate of formation. Okay, some of the results for the DOM characterization. The 3 types of environment recorded very different DOM features. I'm just gonna select some of them because we perform a very deep characterization of the DOM. So briefly, the deep lakes were very low DOC concentration, less than 1 milligram per liter. Also, the streams have very low concentration of DOC, and the shallow lakes were ranged between 1 and maybe 4 milligrams per liter. So all of the environments are oligotrophic environments. The DOM molecular size was measured using the spectral slope, and we have a very low DOM, very low molecular size in deep lakes, intermediate in shallow lakes, and very high molecular dome size in the streams. Regarding the humidification in deep lakes, there was very low humidification. The humidification was measured with the Higgs index, with the fluorescence index. It was very high in shallow lakes, and it was intermediate in streams. So the DOM characterization allowed us to determine different characteristics of the DOM of the different environments. Regarding the natural hydrogen peroxide concentration, we found hydrogen peroxide concentrations, and we also found that there was different concentrations among the environments. The streams have very low concentration of hydrogen peroxide, followed by the shallow lakes, and the deep lakes have a very high concentration of hydrogen peroxide. So this is one of the first record of hydrogen peroxide in the surface water of this environment, and can be considered baseline levels. The hydrogen peroxide concentration is very low compared to other freshwater systems around the world. We are in general less than 200 millimolar, and in some other cases they usually are around 600, 400 millimolar. Okay, about the photochemical hydrogen peroxide formation, we also found that the different environments, the streams, the shallow lakes, and the deep lakes have different formation rates. And the shallow lakes have the highest formation rate, followed by the streams and the deep lakes. And in this plot, I wanted to show you the DOC concentration and the formation rate, and we see 2 clear patterns. This was very wonderful for us. We see a very strong positive relationship between DOC and hydrogen peroxide in shallow lakes and in streams, and we see a negative correlation between DOC and hydrogen peroxide in deep lakes. In a multivariate analysis, we put all of the data of the DOM, and we also put the hydrogen peroxide formation rate, and using 2 axes, the first and the second axis, we can explain almost 80% of the total variance. And as you can see, all of the different type of environments were separated we have the streams, we have the shallow lakes, and we have the deep lakes that they were completely separated, and they all of them have their own characteristics, their own features. So some final remarks, that natural hydrogen peroxide concentrations were found in the surface water from the three types of environments in Argentina. The hydrogen peroxide concentrations range from 5 to 60 nanomolar. In experimental incubation, the hydrogen peroxide formation was due to the photolysis of the dissolved organic matter. And a positive relationship was found between the DOC concentration and the hydrogen peroxide production rate, was found in the shallow lakes and in the streams. This positive trend, this positive correlation could be related to the high humic compounds in the dissolved organic matter. And the negative correlation between hydrogen peroxide and DOC was found in deep lakes. Possible explanation for this particular trend can be related to the photochemical and the biological weathering that these deep lakes experience. Some of them have very high water retention times. So the photoproduction is related also to the DOM characteristic and the reactivity of this DOM. For example, in Andean Patagonian streams, they act like a pipe, and they're just draining all the terrestrially derived DOM with very low internal processes. And in this environment, the water retention time sometimes is just hours. So this is what I was saying. They have very low DOC concentration, they have a high unification, and they have high molecular weight size DOM. So there's no weathering, there's almost no weathering, they're just pipes. Shallow lakes that have higher DOC concentration, and some of them could have intermediate water retention times. In some cases, there's like half a year. So they have substrate because they have a high DOC, and they have molecular intermediate molecular weight size DOM, which is an indicator that they have some sort of internal processing. Some could be like photochemical weathering, for example. And in contrast, the deep lakes have very internal processing. They have very long water retention times. And sometimes this is estimated 2 years, but in one of the biggest lake, it's also been suggested to have been more than 11 years that is the water in these lakes, the water retention times. So we have the prevalence of lower molecular weight size compound of the DOM. So it's very low DOC concentration, DOC concentration. There's very low DOM, and it's not reactive. So it do not produce much hydrogen peroxide. Okay, thank you for your attention, and I'm happy to answer some questions.
Thanks so much, Patricia. I suggest that anybody who has a question just speaks up.
I have one, Lisette.
Yes.
Thank you for an interesting presentation. I was wondering about the role of light in your central hypothesis, because you hypothesized that there's a link between the hydrogen peroxide produced and the DOC concentration in the lake. And yet it's light dependent, this process. And the more DOC or DOM you have, the less light you have. So in a way, the two are in conflict with each other. So my question would be, do you know the sort of the type of relationship between light and the hydrogen peroxide production? Is that a saturation curve, or how would that look?
Yeah, we actually have in some of the environment in the shallow lakes that we have more DOC concentration. It's around, the highest is around 4 milligrams per liter. So I was also thinking about an inner filter, a sort of inner filter effect that light not. It is not the case. This is not the case in the shallow lakes. The UV radiation, although we have some color in the water, the UV radiation almost reaches the bottom of this. They are very clear water. So we have no color in those, so there's no attenuation of the light. They're very oligotrophic and they're very clear water. Some of them you can see the bottoms especially in the shallow lakes, of course, and not in the deep lakes, of course. But in the deep lakes, the secchi is in the summer times is sometimes 20, 30 meters. So they're very clear water.
UV or PAR that is driving the production?
Excuse me?
Is it UV or PAR light that is driving the production of the hydrogen peroxide?
It is ultraviolet radiation. It's ultraviolet radiation that has a more power to break and to perform ELYSIS of the DOM. Yeah.
Thank you.
If there's no more questions, I suggest, because in lieu of time, that we all move onwards to our respective work.